Hello, welcome to the channel Life Learn. Today we will discuss about COVID-19 and watch till end to know everything about COVID-19. Hello everyone, today we are going to learn about COVID-19. CO stands for Corona and VI stands for virus and D stands for disease and 19 stands for 2019, the year when coronavirus disease started. Now the timeline about the COVID-19 disease. The first case was detected in Wuhan, China on 8th December 2019, and the first time the virus was named by ICTV, International Committee on Taxonomy of Virus. So they named the virus as SARS-CoV-2, severe acute respiratory syndrome coronavirus 2, and the disease was named by WHO as COVID-19. And after that, on 11th March 2020, WHO declared COVID-19 as a pandemic. We'll talk about the structure of coronavirus. So because it looks like the spike protein, looks like crown, so then the name is coronavirus. So these are the basic components of coronavirus. So the main component of coronavirus is single-stranded RNA genome, and this contains spike protein, nucleocapsid, membrane protein, and envelope protein. So the most important thing which we will discuss in here are spike protein and the single-strand RNA viral genome. So these two are the most important components of this virus. So now let's talk about how this virus enters the human body and infects human body. So suppose this is a coronavirus. It enters through nose and goes into the lungs. So in lungs, how it develops the disease and how it enters the cell we'll discuss now. This is coronavirus. This enters the lungs, and lungs has several alveoli. So in alveoli, suppose this is one cell and this is the membrane, and on membrane there is one receptor protein called S2. The coronavirus binds to S2 receptor and it enters the cell. So now this becomes the factory, the copy factory of the coronavirus, and it hacks all the components of a cell and it makes its own copy using the human cell. So this is, as I said earlier, this is the RNA, and the RNA comes out, single strand RNA, and the single strand RNA makes its own copy using transcription machinery, and it makes several copies of it, and then it is released out of the cell, and then again it infects another cell. So like this it goes on. In this disease, age is a major risk factor. Below 50, more than 80% of cases show only fever, fatigue, and dry coughs, and some are asymptomatic. So around 14% of cases which are above 60 years, they show hypoxia, or sometimes they need ICU beds, and they show little severe symptoms. And higher ages which have other diseases, they show acute respiratory disease, acute cardiac injuries, multiple organ failures. So these are around 5% of cases which show severe symptoms. Now, how this disease is transmitted? Suppose one person is infected and he or she coughs. So usually the coronavirus cannot move in the air, so it moves through small or bigger droplets. So suppose this contains coronavirus. Some are smaller droplets and some are bigger droplets. So smaller droplets go little farther and bigger droplets move less. So usually these droplets move maximum to 1 to 2 meters of distances, and these droplets sometimes fall on surface when we touch them, and then we touch our face and it goes inside our body. And some droplets which stand in air around 3 hours, when we move to that place we inhale those droplets and get infected. And sometimes if we are talking with infected people, these droplets go into our. So how we are going to detect this disease? When a infected person showing some symptoms like high fever, headache or dry cough goes to the doctor prescribes RT-PCR test. So what is RT-PCR test? RT-PCR is reverse transcriptase polymerase chain reaction. First we will know about polymerase chain reaction. Polymerase chain reaction. So polymerase chain reaction is, first this is suppose one DNA strand, and making copies from one DNA there will be 2 DNAs, and from 2 there will be 4, and so on. So this process is called polymerase chain reaction. So till now we can detect only DNA, but we cannot detect RNA. Remember I talked about the virus has RNA. So first we need to make this RNA to DNA. So this process is called reverse transcription, because this is not transcription. Transcription is usually DNA to RNA. So because this is reverse, so we call it reverse transcription. So first we make one copy of DNA, and then from the DNA we make several copies of DNA. So this process is called reverse transcription. So this, after making the several copies of DNA, we can detect using instrument. So apart from RT-PCR test, there is another test which is called rapid antigen test. By now many of us have seen this type of kits. These are called rapid antigen test kits. As I mentioned earlier, coronavirus have the most important 2 things, which are RNA genome and also spike protein. In RT-PCR test we detect RNA genome, but in rapid antigen test we detect spike proteins, which are unique spike proteins for coronavirus. So in this chip there will be 2 kinds of antibodies. These here there will be primary antibodies, and in these 2 regions there will be 2 types of secondary antibodies. So out of these 2 primary antibodies, one detects control antigen and another one detects the spike proteins. So after when we put these antigens, so the antigen binds to these antibodies, and these antibodies move to bind these secondary antibodies. The control antibody binds to this secondary control antibody, and our test antigen having antibody binds to this secondary antibody. So if we have coronavirus antigen, so these two lines will come out. This is control and this is test. But if we don't have coronavirus, only one control will come out. So this is how we test using rapid antigen test. So when we can detect these viruses in our body, suppose this is the time point when we show symptoms. So usually it takes 1 to 5 days to show symptoms after infection of coronavirus, and after showing symptoms we can detect using PCR test or antigen test. So usually until 3 weeks we can detect using PCR test and it will show positive, but after 3 weeks there is a less chance we can to show PCR positive, but we can still show antigen positive test. And if we check antibody test, it goes around 6 weeks. So when we can detect coronavirus in our system, so suppose this is the time point when we show symptoms. Before that that is very unlikely that we can detect through PCR test. So it takes, after infection it takes around 1 to 5 days in most of the cases to show the symptom. During these periods we may not detect RT-PCR test. So after infection we can detect using RT-PCR or antigen test. So RT-PCR test will show negative after 3 weeks. So even after 3 weeks using antigen test it may show positive, but after 3 weeks RT-PCR will not show positive, and after 2 weeks we can find antibody in our system and we can detect these antibodies even after 6 weeks. So what are the treatment options for coronavirus? Well, there are few treatment options for coronavirus. So remember, as I said earlier, coronavirus enters through lungs and next alveoli and then goes to the cell and then goes to the blood system. So in the blood system it activates several cytokines which brings neutrophils, and these neutrophils destroy coronavirus, and along with coronavirus these neutrophils also destroy some of the cells which are present in the lungs. They also destroy these cells, and also these cytokines move to different organs and destroy them. Chloroquine or hydroxychloroquine, these molecules stop entering coronavirus to the cell, and remdesivir stops replication of viral genome, so it cannot replicate and it cannot form more coronaviruses. Convalescent plasma, this kind of plasma are derived from already infected person which have recovered, and these plasma are derived from those person which contains neutralizing antibodies. These neutralizing antibodies bind to coronavirus spike proteins. Suppose these spike proteins are blocked by antibodies, neutralizing antibodies, there will be no free spike proteins which can bind to these cells and enter the cell. And another option is dexamethasone. Dexamethasone is one of the steroids. As I said here, the coronavirus will activate several cytokines, and these cytokines will bring neutrophils to kill these cells. So overactivation of these cytokines will destroy our own body organs. So steroids are used to block these cytokines and rescue our own organs. So these are the treatment options for coronavirus. As we know that prevention is better than cure, so prevention is always the best. So first line prevention is using mask, social distancing and hand hygiene. So if we use mask, there will be less spread of coronavirus and there is a least chance of exposure to coronavirus. So this is the best option for preventing coronavirus. Another option for prevention is vaccination. So here comes again the same infection procedure. So like a spike protein binds to S2 receptor, and after releasing, the antigen presenting cell detects this virus and it digests the virus, and it shows again shows the viral peptide, which is the spike protein, and this spike protein is detected by T helper cells, and T helper cells activate cytotoxic T cells, and cytotoxic T cells kill the re-entry of coronavirus. Simultaneously, the B cell is activated and B cell releases antibodies, which also inhibits the entry of coronavirus. So similar way, this is the natural way of getting the resistance to coronavirus. Similar way, the vaccine is used. So in vaccine, we take these spike proteins and inject to the body, and after injection, the antigen presenting cell takes these antigens and develop the immune system, and when the coronavirus enters our body, these antibodies bind to coronavirus, and also these T cells as memory, they kill coronaviruses. So now we'll talk about the variants. By now everyone must have heard about UK variants, Brazil variants, Indian variants or South African variants. So now we'll talk how these variants are formed and why these variants are formed. Remember, I said two most important things in coronavirus: spike protein and RNA. So the spike proteins bind to receptor of human cell. So this is the spike protein of coronavirus. So in this spike protein, 5 amino acids. Protein has several amino acids. So out of those amino acids, these 5 amino acids which bind to receptors, human S2 receptors, are most important. So because these proteins or amino acids are formed from RNA, so that is the most important thing. So now we'll discuss about the variants. By now everyone must have heard about UK variants, Brazil variants, Indian variants or South African variants. So what are these variants and how these variants are formed? As I mentioned earlier, now we will talk about the variants. By now everyone must have heard about UK variants, Brazil variants, Indian variants or South African variants. So what are these variants and how these variants are formed? So as I said earlier, what are the two most important things of coronavirus? Spike protein and RNA. So spike protein binds to the receptor of human cell, and this spike protein has one domain called receptor binding domain. So suppose this is coronavirus and this is spike protein. I made this spike protein bigger to look better. So this one has ribosome binding domain, and this ribosome binding domain has 5 important amino acids. 1, 2, 3, 4, 5. These 5 most important amino acids are binding to receptors, and the mutation in these 5 amino acids make the variants of coronaviruses. Coronavirus has RNA genome. This RNA contains A, U, G, C as codon. Suppose 3 nucleotide makes 1 codon or 1 amino acid, and change in 1 codon makes a different amino acid. Suppose different amino acid. So when coronavirus enters the cell, it makes its own copy using RNA dependent RNA polymerase. So during this process, there is a chance that in place of 1 codon there will be a replacement of, suppose here A, U, G, C are there. There will be one in place of G, there will be a C. So this will change to another amino acids. So this will sometimes this replacement of codon will not help the virus, so the virus will die. Sometimes these amino acids, change in amino acids will help the virus to bind to the receptor more tightly. So this, suppose this is the receptor binding domain, so change in amino acid will make this contact, so this will enter easily than before. Suppose here we'll make 2 variants, this is cell and this is the receptor, and this is the coronavirus, and this is receptor binding domain, and this is the normal the first one. When we detected the virus was like this, and after the change the virus suppose the receptor binding domain will change to like this. So you can see the binding will be more stronger here than here, so it enters easily into the cell and it infects more and spread more, and this will become more virulent. So it also escapes the vaccines which are already made. Usually, as I said earlier, we take the spike protein and make vaccine. So because we took the spike protein of the normal virus, but now the vaccine is formed, or the antibody which recognize these spike proteins are formed in our body, but because the new virus has a different spike proteins, suppose like this, and this antibody cannot detect this virus, or it will, if it detects, it will detect less efficiently. So this way the new virus will escape our immune system, and this will grow efficiently and infect more. Thank you everyone for watching my video. If I have missed something and you want to know detail about that, please write in the comment box. I'll definitely make another video for that. And if anyone wants to know about something in biology, and I'll definitely make video. Please write those topics in comment box. Please like, share and subscribe and support me to grow more and make more videos for you. Thank you. Bye bye.